everyone, welcome back to Biophilia, your all-time study mate. Today we are going to continue with the chapter of cell cycle and cell division with its part 2 about mitosis. If you like our video and get some help to understand these in a much more better way, then don't forget to like, share and subscribe to our amazing channel Biophilia. So this is a part 2 about this chapter. अगर आपने part 1 नहीं देखा है then let's try to revise it in a quick. But अगर आपको डिटेल में पता करना है then you can surely go and check out the link in the description box and that will be providing you the part 1 of this particular chapter. So let's shortly understand about the part 1. हमने क्या cover किया था? We had started to understand that each and every cell is going to undergo some process that is called as cell cycle. So cell cycle क्या होता है, all the details will be covered in the previous video, so you can go through that. So cell cycle में हमने देखा था that cell cycle is going to be comprised of two particular functions. So the first one is going to be called as interface. हमने interface को किससे link किया था? That would help you to understand. You can get it in the previous video. So interface is going to be almost more than 95% of the time we had discussed about it, and the rest of the time, that is about 5% of the time, is going to be given for the M phase, or you can say the mitotic phase. So ये जो phase होगा, it is approximately 5% of the total time period. फिर हमने ये भी देखा था that the interface is going to be divided into further categories. So जो interface होगा उसमें categories होंगे like G1 phase. फिर G1 phase में क्या क्या events हो रहे थे, we had discussed it in detail in the previous video. Then we had seen about the S phase. Then we continued with the G2 phase, और G2 phase के बाद we had also done about a very interesting part that was about the G0 phase. So G0 phase के बारे में भी we had studied in detail, and I had given you a very good example to remember about the G0 phase as well. So if you want to know the tricks to remember these in a better way, then surely go and check the previous video out. That would surely help you to understand this in a much more better way. फिर हमने इंटरफेस के बाद देखा था about the regulations, और regulations के लिए भी I had related it to a live example. So फिर वहाँ पे हमने regulations के बारे में बात किया, that is going to actually be the checkpoints. So these are nothing but the checkpoints that we studied about, और checkpoints के बाद जब cell ये checkpoints को cross कर लेता है, finally it is going to enter the M phase. So M phase जो होगा उसमें we had studied there are two things, that is Keryokinesis, that is division of the nucleus, and there was Cytokinesis, which is going to be the division of the cytoplasm. अब karyokinesis में there are going to be two types that can occur. So karyokinesis, there can be two. First, it can occur by the process of mitosis, other can be with the process of meiosis. So very importantly, mitosis जो है, it is going to be covered in part 2 of the chapter, whereas meiosis will be covered in the part 3 of this chapter. So stay tuned to understand meiosis, but for now let's understand about the mitosis in this part. So mitosis क्या होता है? Mitosis is going to be a process in which the parent cell is going to divide into the two identical daughter cells. So importantly, it is going to be the word that is identical, because जब आप meiosis पढ़ोगे, this identical word is going to be replaced. So it is very important to understand mitosis में identical cells बनेंगे after the division of the parent cell. So whatever cells are going to be present, these will be identified, it will be similar to the case of two daughter cells, as well as they will be identical to the cells that are formed. यानी एक parent cell, this is your parent cell में splitting हो गई, उसके two daughter cells form हो गए. This is going to be the daughter cell generation. So daughter cells जो होंगे, these are going to be similar in between themselves as well as to the parent. So that is the beauty about mitosis. So since these are going to be just the splitting of the cells, यानी उनमें कोई variations नहीं हो रहे हैं, these are going to be called as equatorial divisions, because you can say that these are going to split into two halves, and this is going to form the two daughter cells, and they will also have the same number of chromosomes. Very important point number 2. The first one जो हमने देखा, that is the identical cell formation. Second जो है same number of chromosomes, because mitosis में ही same number of chromosomes होंगे. If the case of meiosis, the same number term is going to be changed. So we will go further and understand the entire process about mitosis. सो प्रीवियसली हमने माइटोसिस को इक्वेटोरियल डिवीजन के नाम से भी बोला गया, एंड इक्वेटोरियल बिकॉज इट इज गोइंग टू बी द डिवीजन दैट इज गोइंग टू occur गिविंग सेम नंबर ऑफ chromosomes. So equatorial is related to equal number of chromosomes. So equal number of chromosomes है in the parent cell, then in the daughter cells. So that is a equatorial division. अब equatorial division के साथ साथ it is also called as a somatic cell division. So ये उसका दूसरा नाम हो गया, right? And it is called as a somatic cell division because mitosis जो होता है it is generally going to occur in the somatic cells. So somatic cells यानी जो normally body cells होते हैं, like your muscle cells, your gut cells, your liver cells, nerve cells, blood cells, all these are nothing but going to be your somatic cells. So all the cells and except your reproductive cells. So cells में से reproductive cells को अगर आपने minus कर दिया, then you are going to be left out, that is called as somatic cells. So you can write this as a mathematical equation. So all your body cells minus the reproductive cells, you will get the somatic cells. So इतने सारे cells हैं in your body. So gonads में जो cells होंगे like your ovaries or testes, these are not going to have mitosis for formation of the gametes. Gametes बनाने के लिए there is going to be another process that we will see in the next video. But यहाँ पे हम देख रहे हैं about the division that occurs in the somatic cells. So somatic cells में क्या होगा? It is going to be the mitosis. So whatever is going to occur in the gonads में both the divisions can occur. Gonads को अगर mature होना है, if they want to grow in size, then the growth of the size is going to be because of the multiplication of the undifferentiated germ cells, and that is going to be with the help of mitosis. So very important difference आपको याद रखना है कि अगर gonads की growth का अगर point है, या if it is the increase in number of germ cells, then it is going to be mitosis. अगर वो formation of gametes में है, then mitosis is not going to be functional in the case of that. A third reason it is called as a somatic cell division, because if you consider the case of plants, because mitosis जो होता है it is going to occur in both animals as well as in the case of plants. So animals में you understood about the somatic cells, that if you consider about the mitosis in plants, then you will understand that this is occurring in the meristematic tissue. So meristematic tissues होते हैं where these are going to store food, or they can have air present in them so that the plants can float onto water. Then there are going to be mitosis occurring in the leaves so that small leaf can grow into a larger one, the bud can grow into flowers, then fruits can develop. All this is going to happen because of the cell division that takes place by the help of mitosis. So I hope you understood that why mitosis is called as equatorial as well as somatic cell division. अब mechanism of mitosis को देखेंगे. So mechanism of mitosis is going to be a very elaborative process, because it is, you might think कि M phase जो होता है it is a small phase, it doesn't take a lot of time. But since it does not take a lot of time, it does not mean कि वो complicated नहीं है. It is a complicated process and it is going to involve a lot of important changes, and these changes are mainly going to be taking place in the nucleus and some changes in the cytoplasm. सो बेस्ड ऑन दैट यू कैन अंडरस्टैंड कि माइटोसिस जो होगा उसमें दो मेजर इवेंट्स होंगे एंड द टू लोकेशंस आर गोइंग टू बी having the processes called as karyokinesis. As I told you, it is going to be regarding the splitting of nucleus, and cytokinesis, that is going to be splitting of the cytoplasm. So that was about the two events. Now let us discuss about these events in much more detail. So first आता है that is karyokinesis, that is having the word that is karyo, that means nucleus, and kinesis is going to mean movement. So actually it is going to be like एक जो parent nucleus है, the parent nucleus is going to move and it is going to split into two. So that movement, or that separation of the nucleus is nothing but going to be the process that is karyokinesis. So it is going to be a continuous process जहाँ पे parent nucleus two daughter nuclei में split होगा. So there is no pause between the process, but for our understanding कि हम उसको धीरे धीरे करके समझेंगे. So for that reason you are going to give some time between the activities to understand for your convenience. But अगर आपने actual cell cycle में जाके देखने की कोशिश की, so everything will be going on in such a सिस्टमैटिक वे कि आपको शायद फेजेस को डिफ्रेंशिएट करना मुश्किल हो जाए. And because of that reason the mitosis seems to be taking lesser time, but many things are going to occur in that little time. So for our convenience, let us divide it into 4 simple stages and we will study them one by one so that the changes that occurs in these phases will be easier to be understood. So first phase जिसको आप karyokinesis में divide करते हो, it is going to be the prophase, then the metaphase, anaphase and telophase. So these are going to be your 4 phases that you need to study under the topic of karyokinesis. So everyone with me, let's continue to understand about prophase in detail. Prophase में there are going to be certain changes that is going to occur in the nucleus, because prophase जो होता है it is going to be the first phase, or you can say the first stage. The first stage है, that is the reason the word pro means first, and phase, as you know, it is going to be known as stage. So it is the first stage of the nuclear movement. So it is the first stage of karyokinesis. So उसमें सबसे पहले क्या होगा, as you know that in a normal cell, when you consider कि वो अभी तक cell cycle में नहीं enter हुआ है, it is just lying and had not decided anything, उस condition में if you consider about the genetic material, हो सकता है कि वो DNA हो या RNA हो, but generally we consider DNA because we majoritarily study about plants or animals. So वहाँ पे DNA is going to be present in majority. So genetic material DNA होगा, and this DNA is going to be loose, it is going to be in thin strands, it is randomly placed into the nucleus. अब ये जो है एक ऐसा रैंडम प्लेसमेंट है एंड इट इज़ गोइंग टू बी वेरी डिफिकल्ट टू स्प्लिट दिस काइंड ऑफ कॉइल्ड स्ट्रक्चर into two equal halves. अगर इसको इमेजिन करना है, you can take the example of cooked Maggi. अगर आपने मैगी कुक करके रखी है, then that Maggi is going to be very liquidy, और हो सकता है that it is not going to be easy to divide it into two equal halves. हो सकता है कोई मैगी का टुकड़ा ज़्यादा हो गया, कम हो गया. So in the matter of eating, it is not going to actually matter a lot. But अगर cells के DNA में अगर ये ऐसा थोड़ा इधर उधर हो गया, then what is going to happen? There are going to be many genetic defects that can occur into the animal or the human. So that is not allowed in the case of DNA splitting. So it needs to be perfect all the time. So उस टाइम पे आप क्या करोगे, आपने थोड़ी देर मैगी का जो बना के रखा था, यू जस्ट केप्ट इट एज इट इज, और वो जो वेट मैगी थी, वो वेट मैगी अभी क्या हो गई, सारा उसके अंदर का मॉइस्चर अब्जॉर्ब हो गया फ्रॉम द नूडल्स, एंड नाउ इट हैज़ बिकम अ ड्राई मैगी जिसने पूरा का पूरा बाउल का शेप ले लिया. अब ड्राई मैगी कैसी हो गई है, इट हैज़ टुक द शेप ऑफ टुक the shape of what? The bowl or the container that where it was placed. So कंटेनर का शेप ले लिया आपने, उसको डीमोल्ड कर दिया, जैसे केक को आप डीमोल्ड करते हो, केक को डीमोल्ड कर दिया, you cut it into two and that would be easier to divide. So similarly, for that reason the DNA is also going to undergo the condensation process. जो बहुत से ऐसा लूज सा DNA था, इसको ऐसे पूरा चौड़ा मोला नूडल्स को सेपरेट आउट करना इज़ वेरी डिफ़िकल्ट। हो सकता है कि नूडल का कोई पार्ट टूट जाए, समझो DNA का एक पार्ट टूट गया, देन समझो वो जो पार्ट टूट गया हो सकता है दैट इट वाज फॉर मेकिंग ऑफ योर हैंड्स, सो यू माइट थिंक दैट एक नूडल गया, नो प्रॉब्लम, but if one part of DNA is going away, there can be severe defects that can occur into the external or internal features of the body. So उस वजह से this scattered DNA needs to undergo condensation, and once it is undergoing condensation, the DNA strands are going to form chromosomes. What are they going to form? These are forming chromosomes. So finally जब chromosomes form होंगे, it is going to be much more easier for the splitting of the DNA. So for that reason we see that in prophase there is going to be the first event that is going to be called as condensation of chromatin material. So chromatin material is the गीली वाली मैगी, and once it has started the condensation, it has been becoming into a compact mitotic chromosomes. यानी अब आपके पास केक के मोल्ड जैसा मैगी है, and that is much more easier to cut and give it to the two people. So that is the reason the condensation of chromatin will be starting up first. Then next time what is going to happen, as soon as the chromosomes are going to appear, आगे आपने बॉल में से मैगी निकाली, आपने दो हिस्सों में बांट दिया, बट स्टिल द टू पीसेस आर गोइंग टू look like all they are compressed and kept together. So वैसे ही chromosomes भी होंगे, they are going to appear like a ball of wool. जैसे वुल के स्ट्रैंड्स ऐसे एक के ऊपर एक लपेटे हुए रहते हैं, वैसे ही these chromosomes are going to be completely coiled around themselves, and because they look like a ball of wool, this stage, that is prophase, is going to be called as a sperm stage. So this is a very important point to remember with the reference of MCQs, because MCQs में पूछा जाता है that which stage or which phase is called as a sperm stage. So you need to answer that it is going to be the prophase of mitosis. So after this particular events, side by side, so these events were occurring kahan pe? The A point and the B point, these were occurring in the case of nucleus, right? बट इसके साथ साथ न्यूक्लियस के साथ साथ कुछ चेंजेस साइटोप्लाज्म में भी हो रहे हैं, and these are going to be that the centrioles that are present in the animal cells, these are going to start to move towards the opposite poles, because ये centrioles ही होंगे, these are going to spread out the spindle fiber, and then the chromosomes will be pulled away. So for that reasons the centrioles are going to move, समझो वो randomly कहीं पे भी placed होंगे, these are going to go into the respective poles. So likewise the movement will occur into the prophase. Then the centrioles के वहाँ पे assembly of mitotic spindle will start, so that ropes बनाना शुरू हो जाएगा, so that chromosomes can be aligned in the next phase. Then finally the last event that occurs under the prophase, that is going to be when the prophase is at the late stage, that is end of prophase, you will not see the Golgi complex, यानी organelles disappear होना शुरू होंगे, so that there is a lot more space, so that the जो न्यूक्लियस है, न्यूक्लियस एक कोने में होगा. But now since many chromosomes need to align, this particular nucleus is going to separate out, उसका nuclear membrane rupture हो जाएगा, and the entire cell organelles will disappear for some time, and the entire genetic material will be spread out in the cytoplasm. So for that reason, around the end of the prophase, all the organelles like Golgi complex, the endoplasmic reticulum, even the nuclear membrane and nucleolus are going to disappear for a certain period of time. So even in the photographs you can see how these are going to be transiting towards the second phase, that is metaphase. अब मेटाफेज जो होगा इसमें जैसे प्रोफेज में डिसइंटीग्रेशन ऑफ न्यूक्लियर एनवलप स्टार्ट हो चुका था एंड दिस इज गोइंग टू मार्क द बिगनिंग ऑफ द मेटाफेज. So again it is going to be a very important question regarding who is going to mark or what event is going to mark the beginning of the metaphase. So it is nothing but going to be disintegration of the nuclear envelope. जैसे ही disintegration of the nuclear envelope start होगा, you can try to call it as a early metaphase. And till the time जैसे क्रोमैटिन का जो कंडेंसेशन था वो मेटाफेज में आते आते complete कंडेंसेशन हो जाएगा. And till that time the chromosomes are going to be in the thickest and the shortest phase. And since these are thickest and the shortest, इससे ज़्यादा कंडेंसेशन नहीं होगा, and for that reason it is going to be the most easy phase to understand their morphology यानी उनका appearance कैसा है, all that is going to be easily understood. उनका shape क्या है, वो कितने numbers में है, all that can be understood in this particular phase. So again this is going to be a very important question that can be asked to you. So first question, किसके वजह से मेटाफेज है ये आप पता कर सकते हो. सेकेंड, मेटाफेज में आप क्या पढ़ते हो, you are going to study about the chromosomes because these are thickest and the shortest. And the next event that occurs कि माइटोटिक स्पिंडल फॉर्मेशन जो प्रोफेज में स्टार्ट हुआ था वो मेटाफेज में आते आते कंप्लीट हो जाएगा. So this mitotic spindles are going to undergo a phenomenon that is going to bring the chromosomes to align onto a plate. ओके जैसे सेंटर ऑफ द सेल में ये सारे के सारे क्रोमोसोम्स को लाया जाएगा एंड ये लाने की प्रोसेस को क्या बोलते हैं, कंजेशन. So I hope you understood that whatever the chromosomes might be roaming here and there, समझो एक सेल में एक क्रोमोसोम यहाँ पे है, एक क्रोमोसोम यहाँ पे है, एक यहाँ पे, एक यहाँ पे, सब इधर उधर घूम रहे हैं. And you don't want कि these are roaming everywhere all around. You need to be in a straight line so that the splitting can be much more easier. So in this case what is going to happen, ये जो two poles पे centrioles थे, centrioles अपने अपने mitotic spindles को spread out करेंगे and they will connect to all the chromosomes. And after that क्या होगा, the chromosomes are going to be aligned onto a single plate in the center of the cell और वहाँ पे centrioles होंगे and these will be connecting to the particular chromosomes. So this part is going to occur और इन chromosomes को center में लाने के काम को बोलते हैं congestion. And the center plate, the imaginary center plate is going to be called as the metaphasic plate. So metaphase कैसे होता है, so center of the cell or the it is called as the equator. जैसे earth का equator होता है, similarly the plane which is going to be in the center of the cell is going to be the metaphasic plate. Now whatever the microtubules were to be connecting, वो कनेक्ट कहाँ पे होंगे, वो कनेक्ट होंगे onto the kinetochore. अब kinetochore क्या होता है, kinetochore you see here in the diagram, there is going to be one chromosome जैसे S phase में divide हुआ था. So you know that during the G1 phase it was having one chromosome, but अगर S phase में हुआ तो there is going to be one chromosome but two chromatids. So वहाँ पे उसको बोलते हैं sister chromatids. So S phase में ये जो chromosome है, अगर if you try to zoom this particular part, then you come to this diagram over here. So this diagram number 2 is going to help you to understand कि जो sister chromatids attach होते हैं, these are going to attach onto a disc like structure called as kinetochore. So एक kinetochore होगा onto this one little chromatid and the other one will be onto the another chromatid. और ये जो टू डिस्क लाइक स्ट्रक्चर्स है उनके ऊपर the spindle fibers or the microtubules are going to come and attach. So with this, the metaphase, the second phase is going to end over here. The third phase called as anaphase. Anaphase जो होता है यहाँ पे ana का meaning होता है up and stage as you know it is going to be the phase or you can say otherwise right phase को stage है. So that is about the meaning of anaphase. So anaphase में क्या होगा metaphase में you had brought all the chromosomes onto a single plate एक सेंटर में लाके सबको बिठा दिया था just like good kids in the classroom. सब एक लाइन में असेंबली के लिए खड़े हो गए. सिमिलर केस हैपेंड ड्यूरिंग द मेटाफेज. नाउ व्हाट यू वांट टू डू, आपके सामने एक बच्चों की पूरी लाइन खड़ी है. समझो एक रो में 10 बच्चे खड़े हैं एंड यू वांट टू डिवाइड देम इनटू टू ग्रुप्स, ग्रुप ए एंड ग्रुप बी, राइट. सो दिस एग्जाम्पल विल बी वेरी हेल्पफुल फॉर यू. समझो वहाँ पे 10 बच्चे हैं, ये 10 बच्चों को यू वांट टू डिवाइड देम, right. So these 10 kids need to be divided. How will you divide it? You want to divide them in group A and group B. What you will do, take one child, send in group A, take second and likewise you are going to split them. अब यहाँ पे split करना easy हो गया. Why? Because they were standing onto a single line. So similar case happened during the metaphase. अब ये जो splitting का काम है into two groups will be done into the anaphase. So anaphase में the centromere is going to split into two and there will be separation of the daughter chromatids. So these chromatids are now going to be called as chromosomes of the future daughter nuclei, because as I told you कि वहाँ पे एक chromosome पे two chromatids होंगे और टू क्रोमैटिड्स पे देर आर गोइंग टू बी टू काइनेटोकोर्स और ये टू काइनेटोकोर्स पे देर आर गोइंग टू बी माइक्रोट्यूब्यूल्स विच आर गोइंग टू बी अटैच्ड. अब ये तो हो गया मेटाफेज तक. अब एनाफेज में क्या होगा, एनाफेज में देर इज गोइंग टू बी द स्प्लिटिंग एंड ड्यूरिंग द स्प्लिटिंग देर इज गोइंग टू बी दिस कंडीशन. एक साइड पे ये काइनेटोकोर आ गया. लेट मी शो इट विद द सेम कलर सो इट इज इजियर फॉर यू टू अंडरस्टैंड. सो यहाँ पे ये हो गया chromatids, यहाँ पे kinetochore हो गया and the microtubules were attached over here. So here is going to be the splitting, दोनों के बीच में distance create होगा and this distance is going to create the new daughter chromosomes. So this is going to be the daughter chromosome number 1. This is your daughter chromosome number 2. So these are going to make up the future chromosomes of the new daughter cells. So that is going to occur into the anaphase. So अब ये जो माइग्रेशन होगा यानी एक पोल से लेकर सेंटर से पोल्स तक जाने की जो जर्नी होगी उस टाइम पे सिंस दीज आर गोइंग टू बी पुल्ड टूवर्ड्स द पोल्स. सो पोल्स रीजन क्या होता है, दिस रीजन विच इज इन द सेंटर विल बी कॉल्ड एज द इक्वेटर रीजन, जहाँ पे सारे क्रोमोसोम्स आके बैठे हुए थे इन अ स्ट्रेट लाइन ड्यूरिंग द मेटाफेज. अब ये जो होंगे ये इक्वेटर्स से लेकर पोल्स तक जाएंगे. सो पोल्स पे कौन होगा, पोल्स पे होंगे द द सेंट्रियोल्स एंड दीज सेंट्रियोल्स विल बी सेंडिंग आउट माइक्रोट्यूब्यूल्स एंड दे विल बी पुलिंग दीज क्रोमोसोम्स टुवर्ड्स देम. सो धीरे धीरे शॉर्टनिंग होगी एंड दिस शॉर्टनिंग ऑफ द माइक्रोट्यूब्यूल्स विल लीड टू द creation of the gap between the two sister chromatids and finally they will move away from the equatorial plate और वो कहाँ पे पहुँच जाएंगे, they will move towards the poles. So during this pulling of the things what is going to happen, वहाँ पे different shapes तैयार होंगे. So what are the shapes that are going to occur onto this pulling? This pulling is going to be having shapes because there is going to be difference at the location of attachment of the microtubules. So chromosomes जो होते हैं you might have studied about the placements of the centromeres. So ये जो center में जो part होता है this center में part को क्या बोलते हैं, it is going to be called as a centromere. So this centromere is not going to be always in the center. It can occur in the center if it is called as a metacentric. अगर सब metacentric है तो वो center से थोड़ा upwards या downwards होगा, not in the center exactly. अगर वो acrocentric है then you can clearly differentiate the smaller arm and the larger or the longer arm. And telocentric में तो finally it will act at the end of the chromosome. So because of this reasons, the position of the centromere is going to change, and because the position of the centromere is going to change, वहाँ पे पुलिंग के टाइम पे अलग अलग शेप्स होंगे. So these shapes are going to be studied onto this particular phase. So this phase is going to be very important to study the particular shapes of the chromosomes. So अगर metacentric है, so let me write it down for you. So if it is a metacentric chromosome, यहाँ पे centromeres center में है, then the shape of the chromosome is going to be visualized as V. Why? Because suppose for example अगर ये आपका kinetochore है और यहाँ पे you have attached your particular microtubules, and this is going to be your particular chromosome like this. So this represents the V shape of the chromosome. Next जो आता है that is going to be called as submetacentric chromosome. So submetacentric chromosome का कौन सा shape होगा? It is going to be a L shaped chromosome. And if you consider about the acrocentric chromosome, so acrocentric chromosome में the shape is going to be visualized as a J shape. And finally the telocentric जहाँ पे centromere एकदम end पे है, वहाँ पे you can consider the I shape. So these are the letters जिनका shapes you can consider in the study of chromosomes, that is V, L, J and I. So again this is a very important point to remember. MCQs are going to come from this. So this is the position of the centromere and the shape that is going to occur in the anaphase. So जैसे I told you about the metacentric, how the shape is going to occur. Similarly you can try to draw the pulling actions of the chromosomes और आप देख सकते हो how the shapes are going to be seen in this particular phase. Then the last stage of karyokinesis, that is going to be the telophase. Telophase जो होता है, जैसे pulling start हो चुकी थी anaphase में and they were coming towards the poles. So as soon as the chromosomes reach to their respective poles, you will call the start of the telophase. So slowly and steadily जैसे आपका काम हो चुका है, you have divided the two chromosomes into two halves, and as soon as the division occurs, that is the chromosomes reach to the respective poles, the chromosomes जो condense हो चुके थे वो condensation से now they will uncoil themselves, और जो पूरी गाढ़ी मैगी थी आपको वो अच्छी नहीं लगी, आपने डिवीजन कर लिया, उसको वापस पानी डाल के आपने गरम करके you made it into your soupy tasty wet Maggi. So as soon as the division is completed, the chromosomes also are going to uncoil themselves, और वो जैसे पहले थे यानी chromosomes से लेकर वापस वो गंदे से ऊपर नीचे वाली हालत में आके they have got their identity back. So they were thin, they were slender, long and loose. So they got their identity back as soon as the chromosomes got to the respective poles. अब जो प्रोफेज के टाइम पे चीज़ें गायब हुई थी उन्हें वापस लाना भी ज़रूरी है. So during this time, that is telophase, the nucleolus, endoplasmic reticulum, Golgi bodies and all other organelles that were disappeared in the prophase, they will appear into this particular phase. And finally, as soon as the chromosomes are going to uncoil and form the chromatids, they are going to be nuclear envelope formation, और nuclear envelope form हो जाएगा, then again the chromatin cluster is going to be packed into the डब्बे में बंद करके रख दिया, so that again they don't flow away and spread out in the entire cell. So with this we understood how the nucleus is going to divide with the 4 little phases that occurs into it. सो जैसे ही न्यूक्लियस का डिवीजन हो जाएगा, द नेक्स्ट फेज इज गोइंग टू occur, that is going to be cytokinesis. Cytokinesis, cyto is the cytoplasm. So अब cytoplasm divide होगा because dividing the nucleus alone is not going to be sufficient for the daughter cells. Daughter cells को cytoplasm की भी ज़रूरत है. So for that reason even the cytoplasm is going to be divided. So cytoplasm division is going to mark the end of the cell division. So cytoplasm जैसे ही divide हो जाएगा, it will give you the result that is 2 daughter cells. So अब cell organelles जो होंगे that will be present in the cytoplasm, they will distribute themselves randomly into 2 halves and they will go into the 2 daughter cells. हो सकता है that some organelles, for example A जो A daughter cell है और second जो B daughter cell है, हो सकता है that the organelles like ER ये condition में 2 आ गए, इधर एक ही है. हो सकता है माइटोकॉन्ड्रिया इधर बहुत सारे आ गए, इधर 2-3 ही आए. But whatever is going to happen, these will be replenishing themselves as these cells are going to undergo growth. ग्रोथ होगी तो ये दोनों के दोनों कैसे हो जाएंगे, दे विल बी लुकिंग सेम एग्जैक्टली हैविंग एवरीथिंग एज देयर पेरेंट प्लांट. सो जो भी इधर उधर होगा इन द केस ऑफ नंबर ऑफ ऑर्गेनेलीज को मैनेज किया जाएगा once in the growth phase that is going to be occurring in the new daughter cells. So once the organisms have undergone the karyokinesis, sometimes it is possible that it is not followed by cytokinesis. So हमेशा तो ज़रूरी नहीं है कि एक क्लास में सभी बच्चे अच्छे से पढ़ाई करके 90+ मार्क्स लाए, ज़रूरी नहीं है. हो सकता है कि some people are above average, they are bringing 70%, 80%, okay, it's okay. So some cells they decide कि हमने nucleus तो divide कर लिया, okay, but we are not in a mood today to undergo the division of cytoplasm. So cytoplasm बिना divide किए दो nucleus लेके ये लोग बैठे रहेंगे, and that condition wherein a single cell यानी एक ही plasma membrane के अंदर there are going to be more than one nucleus. हो सकता है generally there are 2 nucleus और there can be even more than 2 nucleus कि अगर वो बार बार repeat karyokinesis करे but cytokinesis ना करे, तो उस condition में जहाँ पे there are going to be more than 1 nuclei, that is going to be leading to a condition called as syntium. Syntium condition में यानी एक ही सेल में there are going to be large number of nucleuses. हो सकता है कि 2 ना हो, 2 से ज़्यादा हो, and all that is going to be combined in a single cell under a single plasma membrane. So ये कब होगा, जब कैरियोकाइनेसिस यानी डिवीजन ऑफ न्यूक्लियस के बाद अगर साइटोकाइनेसिस ना हो तो उस कंडीशन में द सिंटियम कंडीशन कैन occur. So that was thing कि अगर cytoplasm divide हो तो that will give you the 2 daughter cells. अगर divide ना हो तो condition को क्या बोलते हैं. So with this we completed the major part of what is going to happen during the division of the cells, that is with the help of mitosis. So इसके पहले केस में we understood कि cytokinesis होगा तो फाइनल रिजल्ट होगा तो 2 daughter cells, अगर नहीं होगा तो क्या होगा. But how is it going to occur if it is considering to divide its cytoplasm? So अगर animal cells है then animal cells are going to undergo cytokinesis by the formation of furrow. Furrow क्या होता है, furrow is going to be a deep groove, यानी बहुत बड़ा एक छीरा लगेगा into this particular cytoplasm, and the furrow is going to continue to grow, and this continuation of growth of the furrow which is going to deepen gradually, it is going to form the 2 daughter cells. So ये जो फरो होता है, ये फरो की जो ग्रोथ होती है वो कैसी होगी, it is going to occur centripetally and ultimately it will join to the plasma membrane and it will divide giving 2 new daughter cells. So centripetally क्या होता है, you very well know this particular term. If you don't know then it's better if you find it out so that you won't forget it the next time. So centripetally furrow deepen होगा, it will divide to give you final 2 daughter cells. Now in the case of plant cells, animal cells और plant cells को हमने अलग अलग क्यों पढ़ा है? Because there is going to be some difference, and the difference is that the plant cells are going to have cell wall. प्लांट सेल के पास होता है सेल वॉल, जबकि अगर एनिमल सेल्स के देखोगे तो एनिमल सेल्स में प्लांट सेल वॉल नहीं होता है. So it is very important to understand कि cell wall जो होता है it is going to be inextensible, and since it is inextensible what is going to happen, वो जो furrow formation है कि वो ऐसा deepen out होके it is going to break off. So that condition is not going to be possible because वहाँ पे कौन है, the zippy papa of the Bollywood movies. This is your daughter, this is the son, which are going to undergo marriage, that is splitting up, but the rigid cell wall is not going to allow, and this rigid cell wall needs to be told to divide in some different way. So कुछ तरीके निकाले गए हैं by the nature so that this rigid cell wall can also undergo division. So with this we understand the reason and how the cytokinesis is going to be differing in the plant cells and the animal cells. So आपको अभी reason तो पता चल गया कि difference क्यों है, because the cell wall is going to be present in the plants. So plant cell में cell wall present है, that is the reason it is going to be different from the case of animal cells. So अब वहाँ पे क्या difference आएगा, there will be formation of phragmoplast. Phragmoplast बनाया जाएगा, and who is going to form it? The Golgi complex is going to form this phragmoplast, और phragmoplast जो होगा it will grow centrifugally. Now if you remember, the difference also comes over here. अगर animal cell की बात करो तो animals के case में the growth of the furrow की growth कैसी थी? Furrow की growth was occurring the centripetally, right? If you don't remember, just go few seconds back and you can understand it and you can remember it for a longer time. So animal cells अगर होंगे जो furrow की growth होगी, वो furrow की growth कैसी होगी? Centripetally होगी, whereas in the case of plant cells it is going to occur centrifugally. So that is second difference that can be observed over here. And then finally, because of the phragmoplast growth, what is going to happen? The cell plate is also going to start to move towards the center of the cell, और central of the cell की तरफ जैसे ही वो move करना शुरू करेगी, it will meet the lateral walls, and once it meets the lateral cell wall, the cell is going to divide into the two halves. So over here फ्रैग्मोप्लास्ट का एक डिफरेंस आता है, सेंट्रीफ्यूगली ग्रो करने का डिफरेंस आता है, and that is going to make the division of cytoplasm different in the case of plant cells. So with this we completed कि mitosis exactly होता कैसा है and what are going to be the differences that occurs in the phases. But अभी हमने जैसे पढ़ लिया mitosis कैसे होता है, but why is it going to occur? So what is the significance of mitosis? Let's understand it. So mitosis की वजह से सबसे पहले there is going to be growth that is going to occur. So जैसे एक fertilized egg होता है, एक fertilized egg बनाएगा the particular zygote. So one cell से आपकी life शुरू हुई थी, that was your zygote, and zygote has now become an adult man or an adult woman. So this change that occurs, that is going to occur because of repeated mitosis. So एक zygote में mitosis बहुत बार होते जाएगा, and then it is going to be giving out into a adult human. The second one is going to be about the maintenance of the cell size. So cell size should be maintained in a very well manner. जो cell size होती है, the recommended size, that should be maintained. अगर वो cell overgrown हो जाता है, then to maintain the proper functions of the body, that is proper surface area to volume ratio maintain होना चाहिए, and for that reason what is going to happen, the overgrown cells are going to undergo division. So overgrown cells divide करेंगे and that will help you to maintain the proper function of the body. The third significance is going to be that how these are going to maintain the chromosome number. So maintenance of the chromosome number, because at the end of the division both the daughter cells will be having same number of chromosomes as that of the parent plant, and for that reason only you are going to call mitosis as the equatorial division. क्या बोलते हो? Equatorial division, because it is going to have, or it is going to maintain the equal number of chromosomes at the start as well as end of the daughter cells. So the fourth point is about repair. So you know that जो आपका स्टमक होता है, स्टमक में इतना ज़्यादा pH वाला एसिड है that it is going to be very deadly for the cells. So these dead cells need to be replaced out. So these old and dead cells will be thrown away by the body, and the replacement level. So all the replacing work will be done with the help of mitosis in the different cells. Then for the reproduction, as I told you about reproduction, the gonads needs to grow. The gonads यानी जो testis है या ovaries है, these needs to grow and these needs to mature so that they can undergo the gamete formation. So the growth and the maturation is going to be because of the mitosis, that is by the help of multiplication. ये हो गया in the case of animals. If you consider the case of plants, then some plants or some lower animals also can have their reproduction or formation of gametes with the help of mitosis. Then the last point about mitosis is going to be, it is going to be healing and regeneration process. क्योंकि अगर आपको कहीं लग जाता है, if you cut somewhere, तो स्किन कट होने के बाद वापस ग्रो होके completely वूंड को हील करती है. तो वो वूंड क्यों हील होता है? जो आपका हाथ कट गया है वो हील हो गया, या जो स्टिच करने के बाद जो हील हो गया, या आपका बोन फ्रैक्चर हो गया, फ्रैक्चर रिपेयर हो गया, all this is going to be occurring because of the mitosis that occurs in the body. So that is all about the significance of mitosis. So 6 significance that tells you that why you should understand about mitosis, because अगर इसमें से एक भी चीज़ अगर doesn't happen in a normal way in your body, then you are going to suffer from some or the other type of disorder. So that why we study about mitosis in detail. So with this we completed all the theory questions about the mitosis. अब हमेशा एग्जाम में सिर्फ और सिर्फ theory questions आए ज़रूरी नहीं होता है. There are going to be some numerical questions as well that you need to solve. So here are some question types that I can tell you to understand about the final examination questions. So first type जो होता है, it is going to be about finding the number of mitotic divisions that are going to be required to form 8 cells. So as I told you that one cell is going to undergo mitosis and it will form 2 cells. अब ये 2 cells आगे जाके बनाएंगे more 2 cells and that will give you your 4 cells. So finally आपके पास यहाँ पे 4 cells आ गए after 3 mitosis, right? तो वैसे आपको करते जाना है till you get finally 8 cells in your container. Because अब यहाँ पे आप इसे काउंट नहीं करोगे. Why won't you count this? You won't count this because ये ही डिवाइड करके ये 2 बना है. अब ये इसको आप काउंट नहीं करोगे. क्यों नहीं करोगे? Because it has split itself. तो अगर आपने सबको काउंट करना शुरू किया, this is 1 2 3 4 5 6 एंड 7, सो यहाँ पे तो आपको 7 मिल गए, बट 7 आंसर सही है नहीं, इट इज रॉन्ग. हाउ मेनी सेल्स फाइनली डू यू हैव? You are going to have only 4 cells, right? So 4 cells is going to be the correct answer. 7 is not going to be the correct answer. So वैसे आपको 8 cells पता करने के लिए, here I can draw you a diagram. So you started with one single cell and finally you are going to have the 8 cells. So 8 cells बनाने के लिए how many mitotic divisions did you need? So this is going to be given out by the dark blue line. So ये जो dark blue line है, it is going to represent you the number of mitotic divisions. सो यहाँ पे तो आपको 8 सेल्स दिए थे टू फॉर्म, so it was easier for you to like draw this diagram and count the number of dark blue lines. but अगर हमने ये 8 cells की जगह पे आपको बोल दिया that you have to prepare or you have to make 64 cells. अब 64 cells आपको लगेगा कि you can try to draw and consider solving this question, but it is going to take a lot of time. So lot of time that you don't have in the exams. So what are you going to do? You are going to be a smart person, and smart persons, what do? They use formulas. So formula आपको दिया जाएगा, and if you consider that formula, it will be very easy for you to find the number of mitotic divisions. So formula होगा ऐसे कि number of mitotic divisions that are required to make x number of cells. So x यहाँ पे क्या है? X over here is going to be the number of cells that you need to form, that is 8. And how many mitotic divisions do you need to form? So number of divisions is going to be equal to x minus 1. So that is nothing but how much? 7. So what is going to be your answer? Answer to the first question is going to be 7. So if you count over here about the dark blue lines को count करो, you will get the answer as 7. So it was very easy for 8 cells, but 64 cells के लिए क्या करोगे? 64 cells के लिए अब आपके पास formula है, so it will be very easy. So here x will become 64 and the number of generations is going to be what? It will be very simple, x minus 1, that is 63. So you will need 63 mitotic divisions to have 64 cells. So now you imagine if you had to draw 63V like these to get the answer, and that would be consuming your time, as well as आपको exam में इतना paper content भी नहीं मिलेगा that you try to draw this entire diagram and consider solving how many mitotic divisions it will take to form 64 cells. So now as you have this little formula, very easy to remember, x number of cells के लिए you need x minus 1 number of mitotic divisions. अब second type of question आता है, that is going to be how many generations are going to be for the mitosis to form 8 cells. So here I have tried to keep the 8 cells constant so that it will be easier for you to understand the difference between the two questions. So आपको क्वेश्चन पूछा जाएगा टाइप 2 का, बट टाइप 2 के क्वेश्चन का भी आपको आंसर मिलेगा, एज वेल एज यू विल गेट द आंसर फॉर द टाइप 1 क्वेश्चन. सो इट इज़ वेरी इंपॉर्टेंट टू अंडरस्टैंड द डिफरेंस बिटवीन द टू क्वेश्चंस. फर्स्ट टाइप का जो क्वेश्चन है, आपको पूछा क्या गया है? टू फाइंड द नंबर ऑफ माइटोटिक डिविजन्स. जबकि आपको टाइप 2 में क्या पूछा गया है? यू आर आस्क्ड अबाउट द जनरेशंस ऑफ माइटोसिस, राइट. सो आपको अगर ऑप्शन में दिख जाए दैट ऑप्शन नंबर 1 is going to be about the answer is 7, second option is 3, third option is going to be, say for example, it is going to be any number, like इस कंडीशंस में इट इज़ गोइंग टू बी वेरी डिफ़िकल्ट फॉर यू, बिकॉज हो सकता है कि एक आंसर जनरेशंस का हो, एक आंसर नंबर का हो, एंड इफ़ यू नो in a hurry, if you read the question wrong, it can happen that your answer goes wrong. So you should not undergo this circumstances. So it is important to read the question, right? So importantly, you have to read this particular question correctly and read it that whether you are asked the number or you are asked the generations. So now let's consider solving this, right? 8 cells banane hai, yahan pe we have the diagram already, and what you are going to do, this is going to be your parental generation. जो आपका first cell है, it is the parental generation. First splitting हुआ, that is first mitosis occurred, that is going to give you the first generation, right? फर्स्ट जनरेशन में फिर से माइटोसिस हुआ, यू गॉट द सेकेंड जनरेशन, एंड फाइनली एट द 8 सेल्स यू आर गोइंग टू गेट द थर्ड जनरेशन. सो सबसे पहले आपको तो बनाना पड़ेगा 8 सेल्स का चार्ट, देन यू हैव टू फाइंड आउट हाउ मेनी जनरेशंस. So 8 cells छोटा number है. Again, if you try to make it more difficult, try to solve it with 64 cells. Keeping the number constant, 64 cells के लिए कितने generations लगेंगे? Again, you have to be smart. And for smartness, you need formula. So here is the formula. If you need to have the number of generations, so number of generations को आप क्या करोगे? You will be naming it as n. So number of generation is going to be n, and for formation of x number of cells, so over here you are going to have x that is equal to 8. So 8 is the number of cells that you are going to produce. और कितने generations में? n number of generations में. So 8 over here is going to be equal to 2 raised to n. So finally, what is the n value? N value is going to be 3. Right? We also got the answer from counting the number of generations. So that was easy for the number 8. But if you consider about the number 64, so if you want to produce 64 cells, then you are going to see that 64 is equal to 2 raised to n, and over here n is going to be equal to 6. Right? So it will be requiring 6 generations to form 64 cells. So with this smart little formula, you can easily very well find out the number of generations that are required, as well as पहले वाले formula से you can find the number of mitotic divisions. So I hope discussion about this particular type of question was helpful for you, and if you liked it useful, then don't forget to like, share and subscribe to Biophilia. And it means a lot because it gives us much more energy to bring you such content that would help you to ace in your exams. So do consider for like, share and subscribe, and also tell to your friends. That would help us to reach out many more, and as with your help you are helping others as well. So do consider it. सो फाइनली जैसे हमने सारे क्वेश्चन टाइप्स डिस्कस कर लिए हैं, नाउ इट्स टाइम दैट यू चेक योर सेल्फ, आपको कितना समझ आया है. सो फॉर दैट रीज़न आई हैव ट्राइड टू प्रिपेयर सम क्वेश्चंस दैट वुड हेल्प यू टू अंडरस्टैंड हाउ मच डिड यू लर्न फ्रॉम दिस पर्टिकुलर वीडियो. So ये वीडियो को देखकर you can answer all of these very easily, and you can share your answers in the comment section down below, so that we can have a healthy competition to understand how many of you exactly knows all of these answers. So if you know all these answers very good, you are on a good race. You can further solve many more questions. अगर आपको कोई question नहीं आ रहा है at this particular time, you can always rewind the video and try to understand that particular concept and then try to solve it. I am sure you will get the answers to these questions. So that was all from my side for helping you out to understand the concept and giving you some questions to try your knowledge. Now it's your turn to take out many books and question papers that you have and try to solve these and other more questions, so that आपका जो concept का जो strong pillar है, it is going to be lasting longer till your exams. So that was all for this video about mitosis. We will come back soon with the next video about meiosis. अगर आपको ये वीडियो अच्छा लगा है, if you got some help to understand the concept, then don't forget to like, share and subscribe to our channel, that is Biophilia. And till the time we meet you back soon, keep studying, keep smiling, always stay motivated. That's all from my side today. Till then, bye bye. Thank you.